So I'm going to talk today about some experiments we've done recently looking at how microRNAs, how they work and control genes. So I'll start with just background information about microRNAs, so what they are. So microRNAs are an abundant class of these small RNAs, typically only 21 or 22 nucleotides long, that regulate gene expression post-transcriptionally. So they act directly on message RNAs. And so I say they're abundant. A typical mammalian, what I mean is a typical mammalian cell has somewhere on the order of about 100,000 microRNA molecules, which is roughly on par with the number of total message RNA molecules. Close to it. Right. And at this point, it's turned out that over 1,000 unique microRNAs have been identified in the human genome, which means there are as many microRNAs as there are flavors of microRNAs as there are total transcription factors, and there are more microRNAs or unique microRNAs than there are even GPCRs. A really striking and surprising result was done with phylogenetic analysis looking at conservation of microRNA target sites. And it turns out that more than half of the protein coding genes in the human genome have within them at least one evolutionarily conserved microRNA binding site. And so what's more importantly, probably what's being found is that there's a growing list of diverse biological processes which specific microRNAs are found to be critical components of. And so some of them are listed here. And so you see, basically, you see a paper probably once a month or some in Science or Nature where some microRNA is involved in some essential component of some process. And so taken as a whole, but what microRNAs are is there's these little teeny RNAs that as a whole constitute a class of regulatory molecules that have profound effects shaping gene expression in humans. Right, so that's the background. So at this point, a lot is known about where microRNAs come from. They're born in the nucleus, of course. It's these long primary transcripts that have the ability to fold up into a local hairpin structure. And it's recognition of this hairpin structure that leads to a series of processing events that ultimately results in the binding of the mature microRNA, single-stranded little RNA, to an Argonaute protein. And so it's this complex here that's the biologically active form of the microRNA. This complex has many names. Sometimes it's called RISC. But the way that it works is Argonaute uses the sequence information encoded in the microRNA to find complementary sites in message RNAs that are destined to be targeted for repression by the microRNA, just through normal Watson-Crick base pair interactions between the microRNA and the target message RNA. And then once Argonaute parks on the message, it then promotes repression by blocking initiation of translation and also accelerating decay of the message. And so when I first came to Scripps, actually I was primarily focused on this step here, looking at how Dicer catalyzes the final step in microRNA maturation. They worked with Bridget and Clint before they left. But today I'm going to talk about more recent results, looking at this step here. So basically after the complex is formed, and asking really the simple question of how does Argonaute use the microRNA to find its target site? This is actually a really interesting question that I've been curious about for a long time. So I'll talk primarily about this, and at the end I'll talk about more ongoing and more developing work on what happens after Argonaute finds its target site. But I'll start here with just the simple question, how does the microRNA find its target? It's an intriguing question for a couple of different reasons. First, is it turns out that although base pairing between the microRNA and the target RNA is the basis for recognition of the target, you don't need perfect complementarity. You don't have to have a perfect match between the microRNA and the target RNA. And it turns out that some nucleotides in the microRNA are far more important than others for recognizing targets. So for example, nucleotide 1 from the 5' end of the microRNA, it doesn't seem to matter at all what it is in terms of recognizing the target RNA. In contrast, nucleotides 2 through 8, which has been termed the seed region, are the most important part. And most microRNA targets actually just match these 7 nucleotides of the microRNA. That's sufficient to recognize the target message and elicit repression. And what's strange is after nucleotide 8, pairing to nucleotides, the rest of the microRNA is rarely conserved. 9, 10, 11, 12 is almost never seen pairing potential between the microRNA and the target. There is, in a subset of target sites, about 5 to 10% have complementarity to nucleotides 13 through 16 of the microRNA. So it's a second little region. But what's funny is this region alone doesn't seem to have any repressive effect. It can only act to enhance repression that's initially, that first happens through the seed region. And then after it's nucleotide 16, there's really no conservation to the microRNA at the target level. And then the third very peculiar feature of microRNA targets is vastly overrepresented is the presence of an adenosine nucleotide, so an A in the target RNA opposite nucleotide 1 of the microRNA. And what's strange is having an A here enhances repression of the messenger RNA independently of the identity of this nucleotide. So you don't have to have a U here to have repression. Simply having an A here enhances repression by a totally unknown mechanism. So there's all of these strange and peculiar pairing requirements between the microRNA and the target that are interesting and curious. But another sort of simpler question that I'm curious about for a long time is just the mechanism of the search process itself. So if you move to a simplest possible situation, we have just the microRNA paired through just the seed region to its target RNA. You can look at this in a more sort of realistic situation, and it looks something like this. This is the microRNA in red, and it's paired here to the target RNA shown in blue. Now, most microRNA target sites are found in the 3' untranslated region of the message RNA. So it's after the stop codon. And so if you look in Wikipedia, the median length for a mammalian 3' UTR is about 700 nucleotides. So for comparison, that would, just to show you, just to compare the size, that's this big. And so somehow this microRNA has to be able to find these 7 base pairs within this long polymer of potential binding sites. But the search is even more complicated than that, because if you think about it, so that's the 3' UTR. The microRNA has to be able to distinguish the target RNA from all the non-target RNAs in the cell. Now, I'll do a quick math here to illustrate a point. If you calculate the probability of taking 7 random nucleotides and asking what's the probability that all 7 will match the 7 nucleotides of a given microRNA, it's about 1 in 13,000. So it's very unlikely that any given non-target RNA is going to randomly have a recognition site for the microRNA. However, if you allow even a single mismatch at any one of the positions in this seed region, it's asked for 6 out of 7, the odds are about 1 in 700. So odds are you take any given messenger RNA, it will have at least one close match on average, or 6 out of 7 match the seed region. If you go one step further and say we'll allow 2 mismatches, now it's about 1 in 50. So now the microRNA has to distinguish between the target and the non-target. They're both covered in these close matches, and the only real difference is this matches 6 out of 7, while this matches 7 out of 7. And this all has to happen on a biologically relevant time scale. It's the only way you could ever have this happen. It needs to be facilitated. The search process must be facilitated. And so, of course, this is being facilitated by the Argonaut protein. And so our thinking was you can't really understand how microRNA find their targets without taking into consideration the context of the Argonaut protein. So that's what we focused on. So the first thing we did is we asked the question, well, what do these Argonaut proteins even look like, and how do they engage their microRNAs? And so we grew crystals of human Argonaut 2. This is the most abundantly expressed Argonaut protein in humans. There's 4 Argonaut proteins, but Argonaut 2 is the most expressed. And these are the crystals, and we've solved this structure many times now. We have many structures. And so just jumping ahead to the structure here, this is Argonaut protein bound to the microRNA. So Argonaut has these 4 globular domains with these really sort of esoteric names. It's MID, PIWI, PAS, and N. And the important point is that these domains basically make 2 lobes, in between which there's this cleft, and in red is where the microRNA runs. So the microRNA runs through this cleft between these two lobes. And it's almost stretched out in a linear fashion, so that the 5' end of the microRNA is bound here to the mid domain, and the 3' end is bound up here in the PAS domain. It just sort of threads its way through. But you can actually learn a lot, it turns out, by just looking at how Argonaut holds the microRNA. So I'll start by zooming in here at the 5' end. So this here is the nucleotide 1, this is the 5' end of the microRNA. And what you can see right away is nucleotide 1, the base, it's a U here, is actually flipped into this pocket in the mid domain. And it basically makes it impossible in this conformation for this nucleotide to make contacts to any complementary target RNA. So that explains right away why nucleotide 1 isn't really used for recognizing microRNA target sites. Now, in contrast, the seed region, or most of the seed region, nucleotides 2 through 7, are ordered and stacked up against each other. So basically the bases are stacked up against each other with the Watson-Crick faces pointing out towards the bulk solvent. Now, if you move to a surface representation, what you can see is that the way Argonaut holds, oh yeah, sorry, I should say. So right, they're all facing out towards the bulk solvent, but there's also this helix here, this helix 7, which is going to be an important helix. So it's the 7th helix in the primary sequence of Argonaut. It turns out this helix actually sits over nucleotides 6 and 7. And so if I move to a surface representation, you can see that more clearly here. And so now looking at the seed region from this view, what you can see is nucleotides 2 through 5 are splayed out and available for pairing to a target RNA. But then the rest of the seed region, 6, 7, and 8, are tucked inside the protein. And this theme of tucking the RNA inside the protein actually carries through when you look at the rest of the microRNA. So we zoom back out, and then we look at the 3' half of the microRNA. And so we could see nucleotides 12 running up to 21, the 3' end here. And this part of the microRNA is threaded through this channel that runs between the PAS domain and the end domain. It's easier to see if I move to a surface representation like this. So you can see there's this narrow channel that the RNA has to thread through. And the main point, I think the interesting feature here is that this channel is so narrow that interactions with the protein basically disrupt any stacking of adjacent bases in the microRNA. So there's no two bases that stack up against each other. So basically what that means is the protein makes it impossible for the entire 3' half of the microRNA to make interactions with stretches of complementary target RNAs. And so what that means, I guess there you go. There's the nucleotides. And so what that means is that although Argonaut has bound to it this microRNA with 21 nucleotides of potential for binding targets, in this conformation, which we've seen over and over again in crystal structures, only 4 are actually available for making interactions with the complementary messenger RNA. Right, and so that's how Argonaut holds its microRNA generally in the absence of target RNAs. And so once you see this, of course, then you want to know what does Argonaut look like when it actually, what does it look like when it actually binds the target RNA. And so then we went back and we drew crystals of Argonaut bound to the microRNA and the target RNA. So we've got a lot of different structures. Most of the structures we've done so far just use these little target RNAs that just match the seed region. And so I'll talk about sort of generally what this looks like. So looking at, here's a side-by-side comparison. This is Argonaut bound to the microRNA, the first structure I talked about, and then this is the ternary complex, the Argonaut, the microRNA, and the target RNA. So the target RNA is shown here in gold. And so what's nice about having 2 conformations of the same protein is you can interpolate between the 2, and it'll give you a sense of the differences between the 2 conformations. So I'll start, I think it's an easier way to compare structures. I'll start with just the protein alone. So this is the structure of the protein, or the conformation of the protein when the target's not bound. If I interpolate to the target-bound structure, you can see it's a pretty simple conformational change, basically just a rigid body movement of the PAS domain here. The PAS domain moves up, and that makes this cleft wider. Right? If you can do the same thing if you put the microRNA back on, and you can see what happens to the microRNA between these 2 structures. And the RNA actually goes through a much more dynamic rearrangement. You can see there's a huge rearrangement in the 3' half of the microRNA. But also if you look here in the seed region, you can see it sort of breathing. As it breathes, it actually moves more towards A form when the target RNA binds. And so I'm going to zoom in on the seed region here to show what I think is happening. So, okay, just to orient you. Here we are. This is the seed region. This is nucleotides 2, 3, 4, and 5, which are exposed, available for pairing to targets. And then 6 and 7 are occluded by helix 7 here. And I can show this more clearly if I dock onto this structure the actual target RNA. You can see it binds, perfect for pairing to these nucleotides, but then you can see it's going to run into helix 7 here. And so what I'll do now is I'll interpolate to the actual structure where the target RNA is bound, and you'll see as helix 7 moves out of the way, and it actually relieves constraints that it's placing on the second half of the seed region, and then the rest of the seed region actually becomes ordered. So I do that, you can see helix 7 moves, it becomes ordered, and now you have space for the target RNA to come in and make space pairing actions. And so we knew that helix 7 was going to have to move out of the way to make space for the target to come in, because you can't have 2 atoms in the same place at the same time. So it wasn't really a surprise to see this movement. But what was a surprise is what helix 7 does after it moves. It turns out that helix 7 now lies in the minor groove that's formed of the duplex that's formed between the target RNA here and the microRNA here. It's actually easier to see if you move to a side view. You can see here. So this is the duplex here. This is the minor groove. And you can see helix 7 has these hydrophobic residues that make just these really very close, tight interactions with the minor groove. And so this led to the idea that we have. I really like this idea. And that helix 7 actually serves a proofreading function. And so if you think about it, Argonaut has this problem. It has to be able to know when it's bound to the correct target sequence, but it has to be able to do that independently of the actual sequence, because you want to be able to work with any microRNA of any sequence. And so you have to know when you're bound to the right place, but do it in a sequence-independent manner. And the way I think it does is it interrogates the structure of the duplex that's formed when the microRNA pairs to its target. So the idea is, if you have perfect complementarity, as we do here, between the microRNA and the target, you form an A-form helix, which then presents an A-form minor groove to helix 7. You have this nice docking, and it docks up against its minor groove, and you have the stable interaction. If, however, you have a mismatch in the seed region, or even a GU wobble base pair, the bases will slide past each other, and that then presents a distorted minor groove to helix 7, which then doesn't have this nice binding surface, and then is more likely to collapse back down into the RNA, and that'll help displace the mispaired RNA, and also push the microRNA back into its more scanning mode, so it can actually go and find the correct target sequence. So that's what's happening in the seed region between these two conformations. If you remember now, there's also these large conformational changes that happen in the 3' end. And the way I think this happens is it turns out that helix 7 here, which has to move to accommodate a target RNA, is actually part of this rigid body of the PAS domain, even though they're colored differently. So you can see they move together. So when helix 7 moves to accommodate the target RNA, the PAS domain moves as well. And when the PAS domain moves, it opens up it widens this narrow channel between the PAS and end domain. This is easier to see. Again, if we go back to a surface, so here we are closed down. Now we have stable pairing here. Helix 7 moves, pushes the PAS domain, and everything opens up. Now this is really nice, because if you look here in the RNA, so nucleotides 13, 14, 15, and 16, which we know it's a supplemental region for identifying target sites. In the first conformation, the bases are all splayed apart from each other. There's no way you can really make stable contacts to a target RNA in this conformation. But once it opens up, this allows the microRNA to rearrange itself. And now these same nucleotides collapse down in, and you can see they form what looks almost like a little mini seed region, where the nucleotides are all stacked up against, the bases are all stacked up against each other, and the Watson-Crick faces are pointing out. So it's like a second seed region that forms, but only after pairing to the first seed region. It explains why you have to have this requirement for stable seed pairing before the supplemental region can contribute. And so this is really nice, right? Because what this means is, in the first structure, we had 4 nucleotides available for pairing to a target RNA. In the second structure, we have 11. We have the full seed region, nucleotides 2 through 8, and then we also have the supplemental region, 13, 14, 15, and 16. And so the way that I imagine that it works is when Argonaut really binds onto a target, is the target RNA comes in like this, that we know that we have this crystal structure, and it pairing from 2 all the way to 8. And then at nucleotide 9, it turns out in this conformation of Argonaut, the cleft is too narrow to allow the target RNA to base pair with nucleotide 9. In fact, we've done experiments where if you make a base pair to nucleotide 9, it actually reduces the affinity of the target RNA. So it's actually worse to make that base pair in terms of affinity. So I think what happens a lot of the time is, most of the time, is the target RNA actually exits the central cleft. And that's enough to have binding. But if you have complementarity to these nucleotides here, you can then form additional stable interactions through the seed region. This is just modeled, but I think this is probably more or less what it's going to look like. Right? And so when you take these things together, in terms of finding a target site, it leads us to start thinking about this sort of stepwise mechanism for targeting, where initially Argonaut's just using the first few nucleotides of the seed region to look for potential matching sites. And then once it finds that, it then interrogates the full seed region using helix 7. And then once you have this stable interaction, you can then, if it's available, have stable pairing, additional pairing to the supplemental region. This is a really nice model, I think, and it's very attractive. And of course, once you have something like this, you want to test it. And so luckily, at the time, we were also collaborating with a single molecule lab at Truman Ju, who's at Delft University, who was developing a single molecule assay for observing the binding of Argonaut to its target sites. And so the way the assay works is it's a pretty standard single molecule assay, where you have you have a target RNA with a Cy5 dye, which is immobilized on this surface using biotin. Right? And then you enter into this chamber Argonaut bound to a microRNA that has a Cy3 dye. And so what happens is, if Argonaut binds to the target site, it brings the Cy3 dye into close proximity to the Cy5 dye, and you have FRET, which you can then observe by turf microscopy. So a typical result from these experiments looks something like this. This is a time trace where you're seeing no binding events, and then all of a sudden Argonaut arrives when you see this, this quick increase in the fret signal, and Argonaut leaves, and the signal's gone. You can see multiple binding events. And so once he had the system worked out, he could then ask this question of, is this step, you can just test it. So they did a whole bunch of interactions. So basically, what they did is, so this is the microRNA, and then this is the complementary target RNA. As you can see, if you have no base pairing, you get no signal. Well, a really interesting and sort of striking result was if they had even 3 nucleotides, or 3 base pairs of complementarity this is nucleotides 2, 3, and 4 of the seed region here. They started to see these very quick, short-lived binding events. So even 3 nucleotides is enough to have a signal. And then as they increased the complementarity, the binding events became longer and longer lived until you had pairing to the full seed region, and now all of a sudden the binding is very long. And then extending it further, of course, just keeps it very long. And so you can then plot, or you can measure the average dwell time that basically the average time it spends on any site as a function of n, the number of base pairs between the micron and the target. And you can see it's very slow, they're very short-lived in the beginning, and then once you have full seed pairing and you, going back to the structure, engage that helix 7, now you have a very stable situation. So it's clear that the dwell time, the time Argonaut spends on its target site is proportional, not proportional, but is related to the number of base pairs. But what was interesting is the on rate, or the frequency of these binding events, it turns out is not. So if you look at how often these binding events happen, the short ones or the long ones, the short ones happen just as often as the long ones. So plotting the frequency of binding events as a function of n, it's independent. And so this is consistent with the idea that it first just uses those first few nucleotides to find the site, and then it pairs further on. And we actually know it is specifically these 3 nucleotides, or 3 or 4 nucleotides. Because if you move the region of complementarity further down to the seed region, so this is pairing to 5, 6, and 7, which we know are included by helix 7, then the binding rate drops to near background level. So it really has to be the front end of the seed region to make these interactions. And so all of these observations lead to a really nice model for how Argonaut finds its target site. So it's very satisfying to me because what it shapes up to is basically it looks like Argonaut uses what's equivalent to a computational string search, which is the most efficient way to find a pattern in the absence of any other information. And actually, you know this. I mean, my kids know this intuitively. I think everyone knows this intuitively. So I did this experiment in the lab. I gave this to different people in the lab. This is a word search. You have to find the word elephant. And so I gave it to the different people I brought in my office, and okay, try and find the word elephant. And so some people were fast, they found it in 2 seconds, and others took longer. They took 20 seconds. But when I asked them how they did it, the answer was always the same. They always looked for a small motif. First, some people looked for EL, some looked for just E, some looked for PH, because PH is kind of rare. But they all look for a small motif and then search the surrounding regions. This is basically analogous to what Argonaut does, and this is how string searches work. And so that's where elephant is if you haven't found it yet. So, right, so it's like Argonaut. But remember, an important feature of Argonaut is that initially, during the initial search, it sequesters most of the microRNA, the guide. And only allows base pairing to 3 or maybe 4 nucleotides. And I think this is really important because what that effectively does is it limits the time it spends on not close but non-targets. And I can show you can illustrate that, the importance of this feature, is if you do a different word search. I gave this one to the people in my lab immediately after the first word search. So they were already trained on the first one, but they're on average 7.5 times slower at finding elephant in this one. And the reason is because there are so many more near matches to check. And everyone, on average, some people didn't find it actually. They thought this was elephant, but it's not because they're switched. Elephant's here. Right. Okay. Right. So I think this is the essence of how Argonaut finds its target sites. Right. Okay. So now this is how the seed region works in the searching, and then this is supplemental region works. And then the last question about microRNA target sites was this enigma. We just submitted this paper. This enigma of the A1. This is if a target RNA has an adenosine nucleotide, opposite nucleotide, one of the microRNA, it somehow enhances repression. It can enhance repression substantially. And so, but how this works, no one knows. So we then crystallize, of course, Argonaut with a target RNA that had an A in this position. And so just to orient you again here, so here's nucleotide 1 of the microRNA. It's tucked into this pocket. And then base pairing between the microRNA and the target begins at nucleotide 2 and extends down here. And so this is the adenosine that if you were drawing it out, you would say this pairs to this. But of course, because this is a U. But they don't. The adenosine actually slips into this narrow pocket here on the protein surface. You can see it more easily if I zoom in like this. So what you can see is the adenosine binds here. And so what's really nice about this A is the way that it binds, as soon as we saw it, the geometry is very suggestive because basically this A slots in this pocket and it's essentially orthogonal to the direction of the rest of the target RNA as it extends down into Argonaut. And so right away, the idea was that it actually could serve as a physical block if it bound here against Argonaut sliding off of target sites that it's found, if it's found the correct target site. So that was our initial idea. But for that to be true, of course, the pocket has to be specific for A nucleotides. It wouldn't work if it bound all the other nucleotides. And so we went back and we checked. When I say we, I should say this is all done by one graduate student, Nikki Shirley, who's sitting right there. But I was there and it was good. Right, so what we did is we crystallized Argonaut with different nucleotides in the T1 position. So here's an A and a G and a C and a U. And then this is the actual electron density map, show this wire mesh. This is the actual data. And you can see we saw clear data only for the A, well, the G is a little bit of density, but it's not well ordered. And then C is on the outside, maybe, and then U is totally disordered. So crystallographically, the pocket appears to be specific for A. And then also, if you look at biochemically, just look at the binding affinity of Argonaut for these different target RNAs, we see having an A there enhances the affinity about 3 or 4-fold compared to the other T1 nucleotides, which seem to be essentially invisible. So we know the pocket is specific for A, but then actually the question of how specificity was achieved actually threw us for a little loop. It was just a little bit of a hard one. So we know Argonaut will only bind A in this position, but the question of why, it was a little bit tricky. So to look at this, we have to zoom way in and get very, very close here. So this is the adenosine nucleotide. Now we stripped away the surface, and so this is the protein here. These are all the protein atoms. This is the A. And when I first saw this, I was really disappointed because I was imagining there was going to be these very clever interactions with the Watson crick or the Hoogstein face of the A. It was going to be very clear how specificity is achieved, but it's not. The only hydrogen bond between the protein and this A is this hydrogen bond between serine 561 and the N6 amine. And this is a perfectly good, reasonable hydrogen bond. You can draw it like this, where basically here's the amine, it donates a hydrogen, and makes a hydrogen bond with the lone pair of the serine. And so this is nice, but it doesn't explain specificity, because if you think about it, this is perfectly fine for binding adenosine, but the serine also has a hydrogen atom. So there's just from this interaction alone, there's no reason to imagine it couldn't make an energetically equivalent hydrogen bond to guanosine, which has a carbonyl in this position, but then it could, this is a hydrogen bond donor, just switches who's donor and who's acceptor. There's really no reason to think that this should be favored over this. And yet we know that this amine actually is one of the key determinant for recognition, because when we do the experiment again, using inosine, basically we take adenosine and we switch the amine for a carbonyl to make inosine, it no longer binds. We have structures, you don't see it there, and you do biochemistry, it doesn't bind. Conversely, if you take guanosine and take the carbonyl and switch it to make, to put an amine here, and that makes an diamine-purine, it does bind. Actually, diamine-purine binds a little bit better than adenosine. And so it's clear that this amine is important, and yet the protein the only contact of the protein is this one non-specific hydrogen bond. So then we say, okay, we'll have to look a little closer. And so normally, of course, in these structures, luckily they were solved at higher resolution, and so we can see a lot of solvent molecules. So we thought, if it's not the protein, it must be the water. And so these are the water molecules. I should say these are the oxygen atoms in the water molecules. We can't actually see the hydrogens. And of course, water can make lots of hydrogen bonds. It's a very good hydrogen bonder. And normally I would have thought that you can't really maintain specificity through water, in hydrogen bonding patterns through water, because they can spin and hydrogen bond in many different ways. But it turns out, I think this is a special situation, because if we strip away all the water molecules, except for these two here, you can see that these two water molecules actually sit in a very special place. They're surrounded by main chain carbonyls of the protein, which are all hydrogen bond acceptors. So I can show it more easily like this. So okay, so starting up here, we have this water here which interacts with these two carbonyls, which only have lone pairs. So these are obligate hydrogen bond acceptors. So in our model, basically, this water molecule has to use both of its hydrogen atoms to make these two hydrogen bonds, which means it only has lone pairs to present to the adjacent water molecule. So this guy now has to use one of his hydrogen atoms to make the interaction, to make the hydrogen bond to this water up here. Of course, the second water has its own carbonyl to deal with, and so it has to use its other hydrogen atom to interact with this carbonyl, and that leaves then only lone pairs for interacting with the amine. So the idea is that the protein is using the main chain carbonyls to basically pull the hydrogen atoms on this water back, and so it only has lone pairs to present, and that would then be perfect for making hydrogen bond to the amine, but then would actually repel the carbonyl on the G. So that's the model. And it's a nice model because it sort of makes chemical sense and it's cute. But you want to test it, of course. And it's actually a tricky thing to test because you can't mutate water. But you can change the structure of water a little bit. And so the experiment we came up with was to mutate it. So we have, in the back of this binding pocket, there's an alanine 481. And so what we did was, the idea was if you change the surface of this binding pocket and change how the waters bind by mutating it to a threonine. I have this backwards. The red part should be here, the green part, but that's fine. Anyways, putting a threonine here actually sterically provides a little bump to this water. And so we made the mutant protein. And then we saw the structure, and what we saw was the structure to the wild type is essentially identical, except this water molecule then becomes disordered. It's lost. The top water molecule is still ordered. But more importantly, the adenosine becomes disordered as well. And what's important point is this threonine is, I think, 5.5 angstroms away from the adenosine. So there's no direct contact here. And yet making this mutation, totally you lose binding crystallographically. And if you look at the binding affinity of the mutant, it now doesn't recognize the target RNA it doesn't prefer the A over the G target. It binds it as if the A isn't there. So this one mutation that perturbs the water structure, then you lose this recognition of the A1. And so that's the story. And so, all right, so that's how the A is recognized, we think, through this water-mediated contacts. And then getting back to the question of the search, we were also curious to know, because remember that we had this searching business where we had Argonaut uses the seed region to find the target RNAs, but also has this A binding site. So we want to know, is binding to the A nucleotide, is that part of the search or not? And so the answer is it's not, actually. And so to figure this out, we know that having the A there increases affinity. We don't know if it helps binding or if it prevents release. And so we had to go back to the single molecule collaborators and ask the question that having an A in this position, does it increase the on rate or off rate? Or decrease the off rate. And so the way they do the experiment is they have in their chamber, it's all set, and then they introduce the complex, and they see how long it takes before you have the first binding event. And they did this for target RNAs that had a U or an A in the T1 position. And basically, when you plot a number of binding events over time, you see that the two target sites are recognized at essentially identical rates. And so having the A here provides no advantage to the initial recognition of the target site. However, if you look at the dwell time, so basically how long Argonaut stays on the target site, you do see a difference. So if you take, this is a histogram of the dwell time. And so basically for the T1U target RNA, if you can fit this to a single exponential decay, and then the average dwell time is about 0.8 of a second. If you look at the binding events of the T1, it actually doesn't fit a single exponential decay. So it suggests there's actually more than one population present. If you assume 2, the numbers work out, so about 80% of the molecules have a dwell time of binding events, have a dwell time similar to having a U there. But then the other remaining 20% have a dwell time that's 10 times longer. And so we don't really know what this means, but my best guess is, for these short-lived targets, not the full seed region, otherwise we couldn't see it. What happens is the A pops in, basically slides in this pocket. It's one of the last things that happens. And it doesn't always happen, but when it does, it substantially increases the dwell time. So it's consistent with this model that we initially had, where it may prevent Argonaut from sliding off of a target site. And that's how they can massively, this one adenosine can massively increase the repressive power of a microRNA target site. Right. So, okay, so we've gone through all this stuff about how Argonaut has its microRNA. It uses the seed region in these special ways to find the target site, and then it can lock onto the target site using a supplemental region on helix 7, and then this T1 adenosine can really lock it on. So it's a very, very stable interaction with its mRNA target. And then the next question that we're starting to work on is, well, what happens then? And so the mechanism for understanding how microRNAs repress their targets is an old and cantankerous issue in the microRNA field. There have been many high-profile papers that have claimed to have solved this question and have been turned out to be completely wrong. And so there is a growing consensus, but it's still not 100% clear how microRNAs exactly work in repressing their target RNAs. And so we're going after this problem, but we're doing it more, I don't think anyone's working this way, more from a structural perspective. And that is, it really, if you just think about the molecules themselves. So this is Argonaut bound to the 3' UTR. But of course, the 3' UTR is only a small fragment of the message RNA. So just for scale, well, you can see what Argonaut looks like when it's bound to the full message RNA. Of course, I don't know what the structure of the message RNA is. But just for scale, this is the same size as it would be. So it's got a 5' cap, a 5' UTR. The open reading frame would be some part right here. And then the 3' UTR is here, and the poly A tail is here. Now, what's amazing about microRNAs and Argonaut is microRNA target sites can function from any position, essentially, I think, any position in the message RNA. Most of them appear in the 3' UTR, but they can be anywhere in the 3' UTR, and that somehow, just appending this small little protein here when compared to the whole complex can then block initiation that's happening up here, and also promote decay, which begins by deadenylation of the poly A tail. And it can do that from any position. And so how this works, I don't think anyone really knows. But there are clues. So it's clear that having multiple Argonaut binding sites, multiple microRNA binding sites is much, much better than one alone, especially if the two microRNA binding sites are within 40 nucleotides of each other. So these two together are worth a lot more than twice one alone. The other thing that's clear is that the best microRNA target sites appear at the ends of the 3' UTR. So these would be near the stop codon here, and then this would be near the beginning of the poly A tail. For some reason, there's something special about these positions, the most repressive, but no one knows why. And so I think the model I like best for how Argonaut works is how microRNAs work is it brings in, it's clear Argonaut brings in other factors to the message RNA. And so one main factor that it binds to is this large unstructured protein named TNRC6. So TNRC6 is almost 2,000 amino acids long. It has no real defined structure, but it's known to interact with Argonaut protein, and it's also known to interact with deadenylases. And so the idea would be that since TNRC6 has no defined shape, it's actually long enough to be able basically to reach pretty much, no matter how the message RNA is folded up, TNRC6 can probably reach that point. And so then we exploring this model, but basically doing it in just small steps. The first question is asking exactly how does Argonaut interact with TNRC6? This is actually a tricky question. So if you look at TNRC6 in the primary sequence, as shown here schematically, there's this GW domain, which is about 1,000 amino acids long, and this is known to be where Argonaut binds. And then this interacts with the silencing factors, like the deadenylases and things. If you look at the primary sequence of this region, what you see is if you can look at it, it's basically completely disordered. And there are no recognizable motifs at all. The only thing that you recognize is there's an abundance of glycine residues, which are shown as these blue Gs, and tryptophan residues, which are shown as these red Ws. And it's odd to have a tryptophan-rich region because tryptophan is the least abundant of all amino acids, and it's hydrophobic. So it's odd to find it in this intrinsically disordered protein. It's also clear from other labs that the tryptophan residues are important for binding Argonaut. But there's no clear consensus motif. The people have tried to find motifs here, but there really isn't any motif that's repeated over and over again that you can recognize by sequence. So we asked a simple question then, is if tryptophans are important for binding, does Argonaut bind free tryptophan? And the answer is clearly yes. So we tell this right away because we tried crystallizing Argonaut, human AGO2 again, in the presence of tryptophan, and right away we saw the crystals grew differently. And so then we've solved now a number of different structures of Argonaut bound to tryptophan. And what we see is we basically mapped out 3 tryptophan binding sites down here in the bottom of the PB domain. And so these are about as far removed as they can be from the action up here in the seed region and finding the target RNA. So binding down here. If I zoom in, you can see tryptophans here basically sit in these adjacent hydrophobic pockets, which are probably easier to see if I move back to a surface representation. So you can see there's tryptophan 1 and 2 and 3. I sort of just roll through them all. What's important here is that the tryptophans all bind with the indole side chain buried in the pocket, and then the main chain atoms poking out. And so it's possible that all 3 of these tryptophans could be on a single polypeptide chain. But, right, so argonaut may bind this sort of tryptophan repeat. But if that's true, then, right, so we numbered the sites in the order of discovery. So this one's found first, and the second and third. So you can then measure, we can hypothesize that argonaut binds this a repeat that are basically 3 tryptophans, but then you have to measure around the surface because the polypeptide chain will have to reach around. And so basically it's 25 angstroms to get from here to here, and then another 25 to get from here to here, which, if you did the math, works out to be about 8 to 14 flexible amino acids, depending on how flexible they are. So now you can ask the question, do you ever see tryptophans, you can go back to the primary sequence and ask the question, do you ever see tryptophans separated by this distance? And now that you know what to look for, the answer is yes. And so it's actually really surprising, because although the sequence looks essentially random, the spacing of the tryptophans is far from random. And so you have basically these sort of islands of these it's not perfectly even, but semi-evenly spaced tryptophan residues. And so our hypothesis is that argonaut interacts with these tryptophan repeats using these tryptophan binding sites, and that then recruits, and that's how it interacts with this protein here. And it's there are many tryptophans, so it's sort of a flexible, different type of mode of binding. And so we're exploring this hypothesis, and it's still really ongoing work. I think it's turning out to be right. I can show you what we have so far. So basically what we've done is we asked the question, are these sites important for binding, for the interaction? So we make very careful mutations in each site, basically change the hydrophobic character. You put a hydrophilic residue, and then basically now it doesn't bind tryptophan anymore. And so we mutate site 1. So the first assay we have is we just express the 2 proteins, express argonaut 2 with a flag tag and TRC6, and then do a co-IP and look and see if you pull down argonaut, you also pull down TRC6. So if you express flag tag 2 and you pull down the wild type protein, then you blot for TRC6 and you get it. A negative control is a mutant that doesn't bind TRC6 that already been found. It turns out to be a mutation in the tryptophan binding site 1, so that's our negative control. If you mutate site 1, our own mutation, by introducing a polar residue into that site, we block binding. Also, if you mutate site 3, we see the same thing. But strangely, we mutate site 2, the tryptophan binding site 2, we found like 5 times. We tried to mess up this site. It always binds. And so for an IP, mutating site 2 doesn't seem to matter. So only site 1 and site 3 matter. But I think site 2 does contribute to the interaction, because if we move to a different assay, we see it. So a second assay that we have is this tethering assay. So basically this happens, it's less direct, but it's happening inside the cell. You express a reporter, a luciferase reporter that has these box B hairpins. It basically allows you to directly tether your argonaut protein to the reporter message RNA. And so the idea is if argonaut's working, it'll recruit TRC6, which will then repress the reporter. You'll see a repression. And then, right, so that's how you do the assay. And so we do this basically looking at the fold repression. If you tether the wild type argonaut onto the message, you get about 4-fold repression compared to no AGO2 alone. So that's our signal. If you mutate any one of the 3 tryptophan binding sites, you see about half a reduction in the repression by a factor of 2. So that suggests that site 2 actually does contribute, even though it doesn't seem to do anything in the IP. If you mutate, if you double mutants, it's very strange. I'm still trying to work out what this means. If you mutate site 1 and site 2, you still have some repression, but it's inhibited. But anytime you mutate site 3 and 1 or 2 or any of the others, you see a full loss. And of course, a triple mutant has no repression at all. So all this points to this model I'm going to present, and it's super hand-waving, but this is where we are. So we have argonaut that binds, and we know it can lock onto the target RNA. And then the idea is that it interacts with these islands of tryptophan residues. And so it can refer, probably through site 3, which I think is the most important for the initial interaction, because it seems to be the most important. All right, and then we'll explain then why, if you have a second argonaut here, all right, that brings the adenolysis. And then so now the adenolysis are close to poly A tail, kind of, and so you can have repression. Right? If you have a second argonaut, it's close. It's actually physically close to this raft, and so that would explain why having 2 near each other is better than 2 separated. But also, if you have a third one up here, this could, if it's having it near the poly A tail, but having the tryptophans down here, This is super hand waving, but this is what I think happens. I want to test it. It could actually start bringing all of these factors together. And we know TRC6 probably has the ability to bind up to 7 argonauts. So you could, in principle, have free tryptophans, which could bind, this is where it gets really tricky, you could bind an argonaut on another messenger RNA molecule. And if you do that, now you bring another argonaut potentially, and that could bring another TRC6 with more deadenylases. And once you have that, then it's just game on, right? Because you can have as many, you can just keep going. It's almost like an old style IP, where you just keep bringing more and more factors in. And now in this model, it doesn't even matter where argonaut is relative to the poly A tail of the cap, because it's all brought together inside the cell. And it looks kind of crazy, but there's actually precedence for this idea. Because if you look in cells, so these are HeLa cells shown here, TRC6 is known to localize to these, they're called GW bodies, these large silencing bodies inside the cells. That's the nucleus here. And these are GW bodies. And you can see AGO2 localizes to the same bodies as TRC6. If you mutate the tryptophan binding sites, any single one of them, you still see co-localization. So that's not enough to break the interaction. But if you move to the double mutants, well, sorry, I should say, if you mutate site 1 and site 2, it doesn't seem to do anything. But anytime you mutate site 3 in a second site, now you lose the interaction. So it actually starts to make sense, but there's a long way to go in understanding. The main problem is we don't really know what the structure of the MRNP is. And if it has a defined structure, and if it changes during these different cycles. And so that's sort of the challenge we're looking at now, but it's a very difficult challenge. So I'm going to stop there and summarize. So really, there's only 2 points the whole talk, right? The first is that the way Argonaut finds its target sites is a stepwise target search, which is the smartest way you can do it. And so, of course, nature found the smartest way. And the other is that multivalent interactions using these tryptophan binding sites with TRC6, the model is that then promotes building these massive silencing complexes, and that's how messenger RNAs are repressed by microRNAs. And then finally, acknowledgements. So I said this is the lab, and there's some photo bomb guy here sneaking in. Always. Right, so I should say all this work that I talked about today was done by 2 talented graduate students, Nikki, Shirley, and Jess, who are sitting right there. But there's also a lot of other talented people who are doing a lot of other things. I should acknowledge Churlman and Stanley, his graduate student who did all the single molecule work and funding. And really, thank you all. I've been really, really happy to have come here. It's been a great place, and I'm very happy with all the colleagues and the support I've had over this time. And I'm just going to stop there and say thanks.